Um, Thank you all for joining the webinar today. I'm here to talk to you about the GDC whole genome sequencing variant calling workflow updates. My name's Bill. I am the Director of User Services at GDC, and I'm here with my colleague, Zanyu.
This is Zhenyu. I'm the GDC Director of Bioinformatics.
Great, thank you. The next thing I'd like to talk to you about is what we're going to talk about today. For the agenda, we're first going to do a quick overview of the whole genome variants in the GDC, just to give some basic information. Then Zenu's going to give some information on the available whole genome sequencing data, the whole genome sequencing pipeline overview. This is where we're going to go in depth in the pipelines. And then at the end, I'm going to give a quick demo on how to download whole genome sequencing data and some information on the documentation and repositories we have to support the whole genome sequencing data. At the very end, we'll take any questions you have. So during the presentation, I would encourage you to ask any questions you may have. You can type it into the Q&A panel. So that's where we're going to look. We might not see it if you put it in the chat panel, but I just wanted to bring to your attention that Q&A and chat are two different panels. So make sure you click in the right one there, Q&A. So just to get started with an overview here, whole genome sequencing data in the GDC, if you're familiar with the whole exome sequencing data, it's a pretty similar pipeline. Tumor and normal samples are aligned separately, and then the resulting alignments are processed with variant caller workflows of different types. So there's the three main types that I'd like to talk about here, and we're going to get much more in detail, but the first type is the point mutations and indels, or as you'll hear us refer to them in the for the rest of the presentation, SSMs or simple somatic mutation. This is just a kind of catch-all for both the point mutations and the smaller indels. This does not include larger chromosomal rearrangements. The next type are copy number variants, and the third type are the structural variants. These would be the larger rearrangements. So there are two whole sets of whole genome sequencing workflow. And the first one is the one that you'll see a lot of in the current portal. It's the existing Sanger whole genome sequencing workflow set. This includes point mutation from the caveman pipeline, indel mutations from the pindel pipeline, copy number variants from the ASCAT NGS pipeline, and structural variants from the BRASS pipeline. Now, the newer, more updated set is listed here, and this includes simple somatic mutations from the GATK4 MUTEC2, VERSCAN2, the SvABA indel, and Strelka 2. And this also, the copy number variants come from GATK4 CNV and Absolute, and the structural variants come from Manta and SvABA. And just to bring to your attention, Manta and Strelka 2 come from the same pipeline as well as SvABA and SvABA indel. So the major changes here is that we're increasing the SSM calling from 2 callers, so that was just Caveman and Pindel, to the 4 that we mentioned before. And this enables us to have a proper variant ensemble and more support for variants that are called from each. This also increases the structural variant calling from 1 to 2. We're adding absolute for better CNV inference. And the Sanger workflow production, just to give you a bit more information on the logistics, is going to be stopped, and the new whole genome sequencing workflows will start across the GDC. So just to give you a bit of a history lesson on the whole genome sequencing variant data in the GDC. So all the way back in 2020, we did a data release number 27. This is when the whole genome sequencing variants were first released from the Sanger workflow set, the first one that I talked about before. Then just recently, at the end of January 2025, this is when the data release 42 was deployed. And this is when whole genome sequence variants were first released from the new GDC workflow set. So because these are two different sets, and there's going to be a transition. I'd like to explain that a little bit. So all data that was processed with the Sanger workflow set, so this is the older set, will be processed with the new GDC workflow set. So there's nothing that we're just stopping at Sanger. We're going to process all of that data with the new workflow set. Um, However, the Sanger processing will stop. So newer data sets will not be processed with the Sanger workflow, but they will be processed with the new GDC workflow. The whole genome sequence processing is currently in progress, and there's a lot of data. It's a large ongoing project, but we want to get the data out to the community as quick as possible. So any pipeline that is completed will be released in the subsequent data release. For the SSMs, if you're familiar with the whole exome sequencing, you know that there's a lot of files that show up downstream here. So to consider one of these pipelines complete, that means that an annotated VCF is available in addition to the raw VCF, the simple somatic mutation. So um, if an annotated VCF is available, we release that data. So each data release, we will release any whole genome sequencing from the new pipeline as we can. So just to give you a little bit of a nice floral diagram here, the green, and this is to give you an idea of the scale of what this project is. The green nodes there are the new pipelines that variants are being released for. The gray ones are the deprecated ones from the Sanger pipeline, and the two yellow ones are the ones that are currently in progress. And all of these today, at least all of the green and yellow ones, Zenyu is going to give a lot more information about. So that being said, I will pass this off to Zenyu. So why don't you take it away?
Thanks, Bill. Next slide. So we currently have more than 15,000 cases with aligned whole genome BAMs available in the portal. In addition, over 5,000 of those cases have Sanger variant clause. While we no longer use Sanger for production, as Bill mentioned, we will gradually release the rest of the completed calls over time. It won't be a complete set, but there will be more than what we currently have. And for the new pipelines, so far we have released this variant call data from 1,000 to up to like 14,000 cases, depends on which pipeline we're talking about. And we plan to continue expanding this set in future releases and with the goal of eventually providing a comprehensive variant call set for all the whole genome cases in the GDC. So eventually we hope, similar to whole exome data, you will see every tumor normal pairs in the GDC, you can find a complete set of the variants. Next slide. Okay, then I will give some details of all the pipelines one by one. First, I want to mention why we're doing this switch. When it's come to the simple somatic mutation calls, you might recall that in the GDC whole exome calling pipeline, we have this ensemble approach that combine calls from 4 different somatic variant callers. This helped us a lot in improving the confidence and coverage across variant types, and we think that will be very helpful for the users. However, for whole genome calling, the Sanger pipeline only provide 1 point mutation caller and 1 indel caller, which couldn't be used from any meaningful ensemble. So that's why we decided to develop and integrate 4 new pipelines into the whole genome workflow, enable proper ensemble across, and provide appropriate high-quality variant calls. For structural variant calling, as many of you may know, the overlaps between different structural variant callers is often quite low. It's not as high as the SSM callers. This has raised our concerns about how to identify like more confident SV set. And we decided, as an answer for that, to develop more pipelines. I think this enables users to choose how you want to use the data. If you are looking for high confidence calls, you can focus on an intersection of the two pipelines. And if you want to maximize the sensitivity with some sacrifice of the accuracy, you may want to looking for their unions. So it's really different user can decide what you want to do with them. And currently, we are not planning to make an ensemble joint call set, but that may be something if user keep asking, we may add new structure variant pipeline and think about that. Now regarding the copy number variation pipelines, we have received ongoing feedbacks about the limitation of the original SCAD pipeline. In response, we are introducing this new workflow using a combination of GATK4 CNV and absolute, which allows more accurate and robust CNV inference, particularly for a complex genomic regions. And finally, I think a very important factor that trigger our pipeline revamping is the computer efficiencies. The previous Sanger pipelines requires a massive amount of computer power, which made scaling to the full GDC whole genome data set a real challenge for us. By switching to these modern tools like Strelka 2, Manta, we significantly reduced the compute requirement. And these new workflows are much more resource efficient, which make it possible for us to process all the GDC data within a reasonable amount of time, something that we have been striving to do for years, and we just couldn't finish all of those with the Sanger pipeline. So those are the main reasons we are doing this major whole genome variant calling pipeline revamp. Next slides. Okay, I will cover individual pipelines from structure variant callings to SSM callings, and finally I will touch the copy number callings. Next slide. Let's start from structure variant callings. As mentioned before, we has now incorporated two different SV callers, SvABA and Manta. Both of these tools generate output in the standard VCF format as well as BATP format. BATP is a specialized TSV format that represents pair and structure breakpoint. We have adopted this for our structure variant output. And if you look at RNA-seq, we also have gene fusion data that has also both VCFs and BATPs. SvABA being the first pipeline not only detects structure variants, but also produce a indel VCF. This VCF is used beyond the SV callings. It's actually fed into the simple somatic mutation ensemble as one independent indel resources, which I will come back in a few minutes. Manta, we use it a little bit differently. In addition to the structure variants, Manta also generate a SSM VCF called candidate VCF, but it's a much larger VCF that contains a lot of potential somatic variants. A lot of those are false positives, as we can see. So instead of treating that VCF as an independent final product, we use it as input for joint Manta Strelka 2 calling. So basically, the Strelka 2 will take this giant candidate variant sets as input, reanalyze them, and filter them down. A small subset of these candidate VCFs from Manta are eventually retained as part of the final Strelka 2 output. This helps us to boost both the quality and coverage of the Strelka 2 calls. Next slide. Now let's talk about the simple somatic mutation calling pipelines. We have tried to structure these pipelines similarly to what we already have for the whole exome. Each pipeline will generate a raw VCF, a normalized left align and annotated VCF, a adequate level map file that's includes result from a single color. We call that adequate level map. On top of that, we also produce 4-color ensemble maps, which comes in 2 forms. One that is unfiltered and another that is filtered and released as the open access. And that's the one we expect most of the users will download and use. For those 4 pipelines, the first one is GATK4 MUTAC2. This is a newer version than the one we use in the whole exome calling, which we have for GATK3. Some of the MUTAC2 results have already been released in the GDC portal. However, we recently identified a data integrity issue. Some of the variants on chromosome 10 and chromosome 20 are missing from these VCFs. So we are actively working on data fixes and expect to release corrected data in one of the upcoming data releases very soon. So if you have downloaded this data, please pay attention to these chromosomes, or you can wait for the new data fixes we will announce in the release note. The second color is SvABA. This is the one identical to the version we currently use for whole exome. It's a familiar and reliable tool. Although the calls have not been released, it's coming soon. Next slide. The third color is Strelka 2. That's a tool developed by Illumina. As just mentioned earlier, we use it as a joint calling mode with Manta. Manta would generate a large calling VCF that is fed as the input of Strelka 2. So Strelka 2 not only call variant from BAM directly, it will also filter down the Manta candidate VCF, which result in a higher quality call set from Strelka 2 output. And the fourth color is not a standalone tool, as just mentioned. It's the indel product of the SvABA structure variant calling pipelines. This helps us to enhance the indel detection in the overall simple somatic variant analysis. Next slide. Thanks. I will talk about calling number pipelines here. We have listed 2 workflows, GATK4 CNV and absolute. However, those aren't 2 parallel workflows. Instead, GATK4 CNV and GATK4 MUTAC2 outputs are dependencies of the absolute workflow. So the absolute is calling on top of GATK4 CNV. Common number inference is very complex, as the same data can be interpreted in multiple different ways. We have consulted with many common number analysis experts in this field, and the general consensus is that automatic CNV detection can work for some degrees from lots of samples. Manual review is sometimes still necessary for a small subset of samples, because the ASCAD or absolute, those 2 tools we have adopted, don't always produce accurate results automatically without manual reviews. That's why you will hear I talk about manual reviews quite often. For GATK4 CNV, we provide two major output. One is the standard non-integer copy number segmentation files. It's commonly referred to as the segment mean. This is similar to what you will find from the original TCGH G19 analysis. All the output we currently have called the DNA copy workflow that's currently available in the GDC. In addition, as I mentioned, the manual review, so what can be reviewed manually, our genomic expert asks us to provide all the intermediate data analysis output in a archive table. This is will be available in the portal as the control access data. And we don't expect most of the general GDC users to consume this file. This is only left for expert manual review purposes. The second workflow, Absolute, built on top of GATK4 CNV, is a well-known tool previously used in TCGA with the SNP6 array data. This is the whole genome version of it. It outputs integer level copy number segmentation files and gene level copy number values. Basically, it is very similar to what we currently have as the SCAD output. And Absolute actually runs in two different steps. Next slide. The first step is called the model generation. It creates multiple possible models based on the data interpretation, and in which most of essential part is the combination of purity and plurality estimate. I will show you next slide what I mean a model. But the next step of this is called model extraction. So one particular model will be selected, and based on its purity and plurality estimate, the final segmentation and gene level copy number values will be extracted. A PDF report is the main output from the model generation step. This PDF files include all the models. So basically, on each page, there will be one model. I will show you in next slide. And by default, the first model in this PDF is used for the GDC automatic extraction. The result will appear in the GDC data portal as Absolute GATK4 CNV R2. That's the name of this pipeline. It's a jargon, but it's pretty long, but that's how we distinguish the input as the GATK4 CNV, and output is automatic extraction. Because it has GATK CNV in it, which hints maybe later we will build Absolute pipeline on top of some other CNV estimation pipelines. We currently don't have it. We only have this one pipeline, GATK CNV, as the input. And later, if expert manual reviewed models, those models become available, we will release what will be called Absolute GATK4 CNV curated. If both curated and automatic results are available in the portal, the curated one will be the one you should use. It's all preferred high quality data. Next slide. I will just show you one example of this model PDF. As I mentioned, each page contains one model, and we have a lot of model inference, but you can see the main difference is the estimate of the purity and plurality. Once this combination is determined, then the tool can generate the corresponding segmentation file out of the inference. Next slide. In addition to the major pipeline I just covered, there are a few additional whole genome analysis results that might be less visible, but still very useful. These are what I normally call them the hidden gems because their results are not individual files that you can find from the file repositories. Instead, they are node properties, query facets, or some values that can show up in individual pages in the GDC data portal. The first set, I call them BAM matrices. If you're looking for information about BAMs, for example, how many reads are there, what's the coverage, what's the insert size, how many reads are hybrid reads that map to multiple chromosomes, etc., that they are all captured as part of the BAM matrix. And we derive them from each aligned BAM files. So not only the whole genomes, but also whole exomes, RNA-seq. And there are properties on the BAM node. And how to access them? You can get them from APIs, or you can use very fast filtering on the GDC portal. Some of those are in the cohort builder, some of those are in the file repositories. You can bring up customer filters to see them. And very importantly, if you open a file page for any individual BAM file in the portal, you will see we recently add a BAM matrix table. The table will contain these magic values that's for your simple consumption for individual files. The next pipeline is microsatellite instability workflow that's derived from a tool called the MSI sensor tool. MSI is very important for some particular type of cancers. And we derive two values, one called MSI status, the other called MSI scores. They are also saved as the properties of the BAM file, so you can expect to see them at the same place as the BAM matrix. That means the API, some of the facet search, and the BAM matrix table. Just note, and we only call these pipelines on the tumor BAM only, so you won't see these values from BAM that's from a normal tissue. Finally, we have tumor purity and ploidy estimates. As I just mentioned, it's naturally a product out of any integer value copy number pipeline, for example, the ASCAT pipeline or the absolute pipeline. Some people may ask why we don't save this as a property of the sample level or adequate level. The reason is because different pipelines, even they see the same BAM file, they may provide different ploidy and purity estimates. As I just mentioned, even for the same pipeline as absolute, different models will have different combination of these values. Because of that, we felt that it was more accurate and transparent to keep these values attached to the segmentation file itself rather than summarizing a single number at the sample level. Next slide. Okay, I will just touch base on two of the newer workflows we are currently developing. One is the tumor mutational burden. This will provide values such as count per megabasis. And the other is the tumor mutation signature pipeline that will extract known mutation signatures from individual samples. That's all I have. I'll pass this to Bill.
Thanks, Inu. And now I'm going to give you a short demo on how to download this whole genome sequencing data. But before we actually get to the demo, I just kind of want to set a goal here for what we're looking to do, and that's to download the following. We're going to download the annotated somatic VCFs and the Manta bedp files from cases with lung cancer. And I underline lung cancer here because that's actually the first thing we're going to need to consider once we get to the demo. But if you've seen some webinars in the past, you've seen the GDC 2.0 workflow. The first step is to build the cohort. The second step is to do something else with it. But today we're going to focus mostly on downloading files. So we're first going to build a cohort. That's where the lung cancer comes in. And that's going to be followed by a download of the cohort data. So let me go ahead and back out here. And here is the GDC data portal, portal.gdc.cancer.gov. Now, the first thing we want to do is build a cohort. So I'll click on Cohort Builder in the top bar here, third from the left. And if you remember, we're looking for patients with lung cancer. So one way we can do that is go to this field down here, the tissue or organ of origin. So we're starting out with 44,736 cases. These are all the cases in the GDC. And we're going to filter that down to a subset that has lung cancer. So we're just going to type lung into this magnifying glass here to search for the values. And here we have 5 values. We're going to choose all of them except for the overlapping lesion of lung. We're just going to go with the non-lesion one. So as you can see at the top here, we have 5,781 cases. These are the ones with lung cancer. And as we enter those, all of these attributes are in this query bar here. So we could go and start downloading files now, but the other thing I want to do is only show us the cases that have this sort of data. So we're going to do that by going down. Right now we're in the General tab. We're going to go down to the Available Data tab. And we're going to want to filter by the workflow type. And there's a lot of workflow types. There's 35 more. So we're first going to filter by the experimental strategy to kind of pare down that list of workflow types. So here's whole genome sequencing. That's what this whole webinar is about. So we'll click on that. And now we have a much more manageable list of workflow types. So we're looking for, if you remember, we'll go back to the PowerPoint, the annotated somatic mutation VCFs, and the Manta BedP files. So we're going to go back and click on the GATK4 MUTEC2 annotation here, and the SvABA indel annotation. Then we also want the Manta BedP files, and every Manta pipeline produces a BedP and a VCF. So we don't need to filter it down by data format or anything. So we'll just click Manta, and here we have all of the cases that have one of these 3 pipeline data. So there's 1,249 cases. So just to keep this around, we're going to save the cohort. We'll call this whole genome sequencing demo. Click save. And now this is saved. And we're going to bring this over to the repository here. So I'm going to click here. We still have this cohort as our active cohort. And if you remember, we filtered down by these 3 workflow types, but this only filters down to the cases that have these workflow types. Right here, there's almost 8,000 files that are associated with these 1,249 cases. So our next step here is going to be to filter down the actual files by their respective workflow. So we'll actually do the same thing we did in the cohort builder, and we'll filter down by experimental strategy, whole genome sequencing, WGS. So now we have significantly fewer files, almost 16,000. And we will filter by workflow type now. So we'll go down to workflow type, expand that, and we'll choose the two annotations, GATK4 MUTEC2 and SvABA indel. And then lastly, we'll choose Manta. But before we choose Manta, actually, let's take these, and what we'll do is we'll just add these all to the cart. So I'll click Add Files to the cart. If you remember, there's 1,673. We'll add them to the cart, and here they are, 1,673 in the cart. So now we'll unselect these two pipelines, or usually there's a reset button up here. Either is fine. We'll choose Manta, and if you remember, we need only the BedP files. So here's data format, and as Zenyu said, with Manta, it produces both a VCF and a BedP. So we'll click on BedP here, and we have 733 files. I'll click and add all these files to the cart. It adds them to the cart. We're at a little over 2,400. And one more thing I want to bring to your attention is that we're not going to actually be downloading these files because this is a public webinar, and these are controlled access data. So if you do want access to any of this controlled data, you'll need to get dbGaP access to the respective project. So if you want data from TCGA, you would get dbGaP access to TCGA. If you wanted it for another project, you would get access to that project. So the next step here, we have a few options. The first one is to go to the cart and download it directly. But I wouldn't actually recommend it here. This is over 12 gigabytes, and as you probably have experienced before, browsers a lot of times cannot handle that size of download. Um, it might lose connection or something, and usually browsers don't have a pick up where we started option. I think not with the GDC. Um, but what I would recommend doing for this one is if you click in download cart and manifest here, this will download a manifest that you can then use alongside the data transfer tool, which can pick up downloads where they're left off, and it's a command line interface. So if you're interested, that's the way to do it. If you're a little more, if you're a developer, you're experienced using APIs, there's another option to download the manifest, extract the IDs, which is the first column, and then use that to do whatever your custom query is with the API. You might download metadata, you might download the files directly, and you might have a special thing you want to do in your pipeline. So all of those options are available. In addition to downloading the actual files, biospecimen, clinical, sample sheet, metadata, this all gives you the information about the files themselves. So this is which patients each is associated with, which specific samples, what's the clinical data associated with the patients, and then all of the different fields that are associated with each of the levels of biospecimen, samples, aliquots, etc. So that is the downloading data demo. And the next thing I'd like to talk to you about is just some resources for information. So this is the GDC documentation site, docs.gdc.cancer.gov. And if you want information on what we just talked about here, you would click on data in the main toolbar here. Then on the left side, you'll see a bunch of different experimental strategies in the GDC. And what we want to do is click on the whole genome sequencing one. This includes a summary of the data that is produced at the GDC. So a lot of what we talked about during this presentation, the information is here, as well as some command line parameters for how these specific pipelines are run. So if you're interested in repeating some of these pipelines with your own data or data that is not in the GDC, this is a good place to start. But a second place that you might want to look is on GitHub. So up here, and I'll bring up the link because it's a GitHub link. So at github.com/NCI-GDC-GDC-workflow-overview, this is where you want to be. This is a an overview of the harmonization workflows. And here, currently, we're working to get as much of this available to the public as possible. So this is all I'm on an incognito mode right now, so I'm not logged in. So this is all open to the public. Um, this has a lot of descriptions of what these workflows do, and then there are um, other things to download here, like the CWLs, which is the workflow language. Some of these do have Docker files if you use that. And I also want to mention that not all of the pipelines um, are included here. As I said before, we're working on getting as many as we can, and there are some various reasons why there might be some roadblocks there. But the GDC works as hard as we can to make this as public as possible. Um, and the last thing that I want to plug real quick is if you're interested in all of this, you'd want to go to gdc.cancer.gov. And if you want to know when we release new data, when something significant is happening with the GDC, when we there might be a maintenance window or something, I'd recommend going to support, and there's an option down here, join the GDC user listserv. And if you choose that, it takes you just a second to put your name and your email address, and you will get a notification for when any data release happens or a software release or anything you as a user of the GDC might need to know. So that being said, that's the end of the presentation, and I want to open it up to any questions. So let's take some of those. Okay, so we have one here. Have you compared the new whole genome sequencing workflow with other whole genome sequencing workflows, such as, I think it's pronounced Wiggets from HMF, to see how similar or different the SSM, CNV, and SV outputs are? Thank you. So Zenu, I'll hand that one off to you.
Yeah, the short answer is no, we haven't compared our workflow output to that particular pipeline. Get more details. Our workflow are not coming out of directly from our imagination. So we do have a genomic data analysis working group, which is a group of scientists who work on a lot of projects. For example, the core analysis person there are the ones did all the TCGA analysis. And we constantly ask them for suggestions and consult them with what tool to use and what your experience with different workflows. And workflows we pick are the ones this team have used or have recommended us to implement. But to answer your question, we haven't compared to the particular workflow you just mentioned.
Thank you, Zenu. I don't see any other questions. We'll give you all a couple minutes to ask any, but yeah. In the meantime, one more thing I'd like to point you all to, since we do have a couple minutes, is if you are interested in some of those different fields that Zenu was talking about, like the BAM fields or the purity, polity, you can go to the repository, and if you click on add a custom filter here, you can see things like MSI score and status, and this will allow you to filter by MSI score or status. You could put something like contamination, and make sure you click on the one that's just contamination, because this is like the downstream files that are associated with the BAM, which is something you might want. But if you're looking for the BAMs themselves, I'd search directly for something like contamination, which you can filter here, or tumor purity. You can search for something like that, and they'll appear um, as cards on the left here that you can filter from. Okay, is there any approximate timeline estimate on the next GDC whole genome sequencing data release? So the next upcoming release, which is going to be data release 43, is being actively developed right now. Um, that that will have another chunk of whole genome sequencing data. It won't have all of it, but there will be more. Um, that's going to be the next data release. It's likely to be sometime in the month of April. Yeah, stay tuned and sign up for the listserv, and you'll know right away. Um, okay. Next question. I saw that you are developing mutation signature tools using Musical. Just curious why Musical over tools like Sig Profiler. Send
Um, um, yeah, initially we try to adopt stick profiler. It's the classic one and developed by Sanger. So it got more frequent update of the new signatures. Um, but we have talked to a lot of experts, as just mentioned, and also did some comparisons and look at um, some of the recent review papers. I think eventually, collectively, we decided the musical as a new tool is the one we want to implement. We did some comparison also. Uh, Also, just want to clarify the mutation signature pipeline. We will only run it as the individual sample non-signature detection. And we won't run in the mode of a cohort level new signal detection. I think we will leave to the users of the GDC to do. But we will extract in the individual level, the individual sample level, what non-signatures are there.
Okay, the next question. So this is from the same person who is asking about the comparing the new whole genome sequencing workflows with other ones. So just as a follow-up, have you assessed the sensitivity and specificity of the new whole genome sequencing workflow, e.g., by benchmarking against reference data sets?
That's a good question. No, we haven't. We will derive um, more data, and then once we have the ensemble map, we might look into how they compare. On the other hand, it's a little bit difficult to compare directly in the existing GDC dataset because there are no known truths there. I think there are some validated dataset from the TCGA that's what we have used for the whole exome calling. If you are interested in that, you can look into some of the papers the GDC have published, including the original paper that compared HG90 and G38 data. That's probably something we will also do once we have collected enough whole genome variant calls. But we have not planned to use like genomic in a bottle data or like a curated sample from 1,000 genome project to do such comparisons.
Thanks, Anu. So here's a question. Where should I get the recording of the meeting? So we will process this recording and give it all the proper 508 compliance things, making sure that we have all the uh, captions and all that. But one way that you will be able to get to it is you go to gdc.cancerga.gov, which is here. You click on go to the support tab here, and then go to the sorry, it went away. Go to GDC webinars on the left here. And for this one, what you'll see, this one isn't there yet, of course, because we're doing it now. But in the past, here are some of the past webinars, and you'll see that there is a YouTube link here. So you can click on this, or you can click read more, and you can also watch this on YouTube. So this will be available on YouTube eventually. Hey, the next question, are all GDC whole genome sequencing data all from short read sequencing?
Um, Currently, yes. We do have some data sources with different data types, and you might also know TCGA also have tried some different data types before, but we don't have pipeline to process this data. So currently, we don't have a plan to process anything other than the short read.
Okay, I get another question here. At the GDC data portal, can I build a data set that has whole genome sequencing patient samples at diagnosis before treatment and at the time of development of resistance/relapse? Um, so we have, I know that whole genome sequencing, most of what we have now was taken at a certain time for for a patient, and I apologize. I don't think I can answer exactly which type of data sets we know of, but what you can do is we have um, a field that you'll likely have to go into custom filters here to look for, and there is a field called um, it is the word procurement, days to sample procurement. So this will tell you when the sample was actually removed from the patient. And typically for most projects, and I would recommend checking the days, the index date field because it's not always the case, but for most patients or for most cases in the GDC, day zero is diagnosis. So if this is negative, um, it is pre-diagnosis, or of course it wouldn't be pre-diagnosis, but the lower number would be um, the lower number would be closer to when the sample was or when the diagnosis happened, and a larger number would be further away from the when the diagnosis happened. And then once you have this information, if you have treatment data, which actually I think we can find in the default fields here, um, if you have, oh, you know what, that will be in the custom field. So if we do days to treatment start and treatment end, this will tell you when all of the different treatments happened. And then there's also days to recurrence and follow-up and diagnosis, and you can find information about that. So I guess the short answer is that we don't have like an out of the box for this specific use case, but the GDC does offer lots of clinical data. Is for if you want to see which sample was collected when and how that relates to all the time points in a patient's history. Okay, are there any more questions? Okay, well, if that is all of the questions, I thank everyone so much for attending this webinar. I appreciate you coming here and learning about whole genome sequencing with all of us. Um, and yeah, just kind of dragging out a little bit to make sure there's no extra questions. Looks like there's not. So thank you all so much. I will stop sharing now. And I hope you all have a good rest of your day. Thank you.
Thanks, everyone.